Richard Hamilton, and I'm currently a PhD student here at San Diego State working in Dr. Kaluzhnia's lab. And kind of what we study in her lab is a specific bacterium. And what this bacterium is it's a methanotroph. And what that methanotroph does is it metabolizes methane as its main source of carbon and uses this methane for biomass or proteins in its metabolism. And kind of why this is such a big picture idea is because there's many anthropogenic sources releasing carbon into the atmosphere every day. And some of these sources are from coal mines or landfills or livestock. And what our idea is to try to take some of these methane sources and feed it to our microbe as its food source. And then why we want to do that is then we can genetically engineer our microbe to produce specified off products or specified proteins of interest, such as spider silk or lipase, or also other metabolites such as ectoin or PHB, which can be used to produce plastics. And what we want to try to do is produce a system that produces proteins or these metabolites in an efficient manner with high yield. And some routes that we can take to do this is we start with the gene of interest. So let's say we're going to take spider silk. And right here I'll just write spider. In order to transcribe this gene, you need a specified promoter to start transcription. And there's many different types of promoters that you can use, and we want to try to steal one from our microbe that has already been shown to show high transcription of the already known genes in its metabolism. And some of these would be something like MXAF or PMO. And one that's a really huge factor that's always being transcribed is this PMO gene. So we want to try to take this promoter region and put it right here, because usually PMO is used for the metabolism of methane to methanol, but we want to try to make it so this promoter factor is transcribing our spider silk protein. And this is one way to increase our protein yield, but also we can go to the next state, which is for translation. And when we go from here to translation, then we also have to think about other factors, because this becomes now amino acids. And we have either alanine or serine. And what usually brings these into the system for translation is these tRNAs. And over here, let's say tRNA. Alanine, let's say, for instance, can be brought to the system either from a codon, which is GCC, or it can be GCA. And there'll be two different tRNAs for this. So let's say for here's one tRNA, and here's the second tRNA. And this one is our GCC, while this is our GCA for our little alanines. And we find that our microbe, it likes to use tRNA B here more efficiently than it likes to use the tRNA A. So we want to try to make sure that this codon sequence, or this sequence, is in our spider silk gene here to be the GCA, instead of having that GCC, so then once again we can have that increased yield of our protein in the overall system. And then to move this even further, we want to, after we have this protein yield, we want to go more into the metabolism of our microbe, where we can take our methane, CH4, and then see how it feeds into the system, and then it goes into different metabolic pathways, and some of them will be offshoot as CO2, or it can come off as glycogen, or it goes into this TCA cycle down here, which then can be used to produce, over here, one of these metabolites as ectane. And we already, in these specific constructs, we have our proteins of interest already to produce the metabolite ectoin, but what we want to do is look at the metabolic flux of the overall system and try to direct the carbon, so most of the carbon is going towards this ectoin end product. So what we can do is either knock out specific genes or downregulate some of these other metabolic pathways to decrease the carbon sinks in that direction. So overall, with this whole system, we want to try to create this efficient production of either metabolites or proteins from methane gas to try to decrease decrease methane emissions overall in the environment and decrease climate change. And thank you for watching my overall talk. Nope.